Well, take 25. Actually, not that many. But we left with this set of links last time. And we're going to talk about blood in relation to genetics real quickly. These are great videos to watch. Mr. Anderson's bloody video is excellent, and I suggest you watch it. I especially want you to take a look at this chart um, that he has, and he talks about how O negative blood is the universal donor, and AB positive blood is the universal recipient. This chart is very useful for determining the type of blood that needs to be given to a person who needs a transfusion. Okay, so understand that. What we're going to do is talk about, not that, this one. Okay, we're going to talk about the types of blood that we have, and there's actually the possibility of 8 blood types. You can have A, which means you have an A antigen on your red blood cells, and antibodies against B antigens. If you have B, you have a B antigen on your red blood cells and antibodies in your blood against A antigens. So if A gets B blood, A's antibodies attack B blood, make it clot, and precipitate out, you get a big anaphylactic response and you die, unless something's done about it pretty quickly. You can also be AB, in which case you're not A, you're not B, you're AB. You have both. That's codominance. Or you can be O, in which case you have neither. Okay? So an AB person has no antibodies against A or against B. Isn't that nice? Okay, so there's AB, AB, and O. Those are the 4 types that we have, and there's basically the 3 allele types, A, B, and AB. 2 allele types, AB and AB, or no allele. No trait, okay? Then you have the RH, RH positive, RH negative. This follows the Mendelian genetics that you can use Punnett squares on. RH positive and RH negative. AB plus is the universal recipient, because if you have the antibodies, you don't have, the antigens, you don't have any antibodies against it, or you'd be killing yourself, okay? Or you can be a RH negative. So O negative is the universal donor, and AB positive is the universal recipient. Okay, so here's the possibilities. The way that Mr. Anderson and others designate it, you can be AI, and I simply means O. It means you don't have the other one. So when the sperm and the egg unite, you can be AI or you can be AA. And at the same time, you could be either plus plus or minus plus or minus minus. If you are plus at all, you're plus. You're RH positive if you have one allele, because here is where we have the dominant and recessive. On this side, we have the co-dominant situation. So for each of these, we have either your RH positive or your RH negative. So RH positive is more abundant, obviously, than RH negative, because you have to be double recessive to be negative. Okay? Okay. So here's where we get down to some of the important questions. Okay? So why do blood antigens kill people, but other antigens from invaders do not? What are antigens? They're foreign substances that get into your system, into your blood, or maybe not into your blood. In the case of blood transfusions, they are antigens that are on the blood cells. And the antibodies are the little soldiers that your white cells, your mast cells have made to fight against the antigens that it doesn't want to see. This is how we fight infection. But it also sometimes is how we get an allergy. Normally an allergy is a mild anaphylaxis, a mild response to a invading substance or protein that gets into our system. And some people think that the anaphylaxis results from being exposed to normally unharmful things when our immune system is bored. I guess that's about the simplest way I can put it. So there is a theory that allergies develop more often in kids that are too clean, that their parents keep them too clean and don't let them get dirty. Used to be a Polish lady, I understand, in Chicago that used to take her kids out once a year in the spring and take a big spoonful of dirt and shove it down their throat, made them eat it. And those kids were always very healthy. Some people thought, she's exposing her kids to all these different allergens while they're young and growing so that they do not develop allergies because they're being exposed to these while they're kids. Maybe so, maybe not. I don't know. It's an idea. So anyway, so the question then, back up here, why do blood antigens kill people but other antigens from invaders do not? Well, sometimes the other ones do. You know, an allergy to a bee sting or a fire ant sting. I had a dog that I thought had died from a fire ant sting one time. Usually what happens is you get an exposure, and then the next time that you get an exposure, you start to develop antibodies against the invader. And then the next time, let's say that this spring there's a lot of pine pollen out and I start to be a little bit irritated by it. Well, then it goes away after a month or a few weeks or whatever. Then next year we have a lot of pine pollen again. Now I have a severe reaction because my mast cells have already set up to produce all these antibodies against the invading pollen. But anaphylaxis does kill people, and it's not always mild. Think of the peanut butter allergies that some children have, and that's why we don't really want peanut butter in schools anymore, because we don't always know what child is going to have a severe reaction and possibly die if we can't get them to the hospital fast enough. Of course, we use epinephrine pens to fight against these reactions because they minimize the swelling and so forth that goes along with it. But why do blood antigens normally kill people? Why are the transfusions with the wrong blood very serious compared to most allergies when it's basically an antigen-antibody response? Well, it's because it's a severe anaphylaxis. Because you've dumped a lot of blood. It's not just a little bit. It's not just a little bee sting. It's not just a little bit of pollen in the air that you breathe. You've dumped a lot of blood into their system all at one time. And what happens is that the antibodies cause these blood cells to clump up and block blood vessels. So you get more than the typical anaphylaxis, and if something isn't done quite rapidly in a hospital for somebody who got the wrong blood, then they're likely to die from it. So it's a serious situation, and it's a very serious business to have the right type of blood for a recipient if they really need it. And of course, as Mr. Anderson says, O negative is really sought after by hospitals because it means that you can give it to anybody. And AB positive people are pretty rare. So, you know, usually you have to match the blood up. And So that's why, in part, hospitals are looking for it all the time, as well as the fact that it doesn't last forever. It has a shelf life. Okay, so that's pretty serious business. But that's the genetics behind blood alleles and blood types. And so you look again at Mr. Anderson's video, look at your textbook and read it, look it up on the internet, and try to understand the genetics here. This is one of those things that just doesn't follow the Mendelian genetics. It's much more complicated, and so it relates to question number 4 in your lab. When do Punnett squares not work, or when are they not useful for determining the distribution of alleles? Thanks, and have a good week.